Chapter 10: Preventing Infection at Mucosal Surfaces. The communication functions of mucosal surfaces render them vulnerable to infection. The mucosal tissues of the human body perform essential communication functions with the external environment, functions like breathing, eating, reproduction, and waste elimination. But this accessibility also makes them highly vulnerable to infection. This figure maps the key mucosal surfaces: the respiratory tract in blue, the gastrointestinal tract in red, and the urogenital tract in green. It also includes mucosal tissues like the lacrimal and salivary glands, mammary glands, and conjunctiva. Together, these surfaces account for over 3/4 of the body's immune activity, making mucosal immunity a critical line of defense. Mucins are gigantic glycoproteins that endow the mucus with the properties to protect epithelial surfaces. Mucins are giant glycoproteins that make mucus an effective protective barrier for epithelial surfaces. This figure shows the structure of a secreted polymeric mucin molecule. Each mucin is made of a single polypeptide chain with N-terminal and C-terminal globular domains. These domains enable mucins to form complex, branching polymers. The dense sugar chains attached to the mucin backbone attract water and trap pathogens, forming a hydrated gel that lubricates and shields the epithelial tissue from damage and microbial invasion. This chart summarizes the different mucin polypeptides, their genetic locations, and where they function in the body. Mucins are either secreted or membrane-bound. Secreted mucins like MUC2, MUC5AC, and MUC5B are produced in tissues such as the colon, airways, and salivary glands, helping trap pathogens and facilitate mucus flow. Membrane-bound mucins like MUC1 and MUC4 are expressed in tissues like the breast, pancreas, and airway epithelium, where they serve as a protective barrier and signaling platform. Together, these mucins form a dynamic network that safeguards mucosal surfaces throughout the body. Commensal microorganisms assist the gut in digesting food and maintaining health. This diagram highlights the major structures of the human digestive tract, from the mouth to the anus. Along this path, commensal microorganisms play a vital role in maintaining gut health. In the small and large intestines, trillions of beneficial microbes aid in breaking down complex carbohydrates, synthesizing essential vitamins, and defending against harmful pathogens. They also help train the immune system to tolerate harmless antigens. Together, the digestive organs and the microbiome form a powerful partnership essential for digestion, nutrient absorption, and immune homeostasis. Commensal microorganisms in the human gut perform 5 essential functions that support our health. First, they synthesize essential metabolites like vitamin K, which is required for blood clotting. Second, they break down plant fibers, releasing short-chain fatty acids used for metabolism and biosynthesis. Third, they inactivate toxic substances from food and pathogens, converting them into harmless compounds usable by human cells. Fourth, they prevent pathogens from thriving by limiting their growth and access to resources. Finally, commensals interact with the gut epithelium, triggering the formation of gut-associated lymphoid tissue that strengthens the immune system. Together, these actions highlight the vital partnership between our body and its microbial inhabitants. The gastrointestinal tract is invested with distinctive secondary lymphoid tissues. The entrance to the gut and airway is protected by a specialized ring of secondary lymphoid tissues known as Waldeyer's ring. This immune ring includes the adenoids, palatine tonsils, and lingual tonsils. Together, these tissues form the first line of immune surveillance, ready to detect and respond to pathogens entering through the mouth or nose. This anatomical arrangement ensures that immune responses are rapidly initiated right at the gateway to the gastrointestinal and respiratory tracts. Deep within the lining of the small intestine lies an intricate network of immune defenses. This diagram shows a cross-section of the intestinal wall, highlighting Peyer's patches, isolated lymphoid follicles, and M cells, all of which contribute to immune surveillance. Antigens from the intestinal lumen are transported across M cells to Peyer's patches, where immune cells initiate targeted responses. From there, signals are relayed through lymphatic vessels to mesenteric lymph nodes, ensuring that the gut maintains both tolerance and protection. Together, these structures form the foundation of the gut-associated lymphoid tissue, or GALT. This histological section shows the organized architecture of a Peyer's patch embedded within the intestinal wall. To the left, the villi absorb nutrients, while just beneath the surface lies the dome region, where antigens from the gut lumen are sampled. Beneath the dome, we find the T cell area, where helper T cells coordinate immune responses. To the right, the germinal center, or GC, marks the site of B cell proliferation and antibody production. Together, these regions form a powerful defense network, ready to respond to pathogens while preserving tolerance to beneficial microbes. When the gastrointestinal tract lacks exposure to commensal microorganisms, we observe both anatomical and immunological consequences. Anatomically, the cecum becomes enlarged, the small intestine lengthens, and lymphoid organs like the mesenteric lymph nodes, Peyer's patches, and spleen are significantly underdeveloped or reduced. Immunologically, there's a notable decline in secretory IgA and serum immunoglobulin levels. T cell counts drop, and their activation is impaired, especially in mucosal tissues. CD8 T cells lose cytotoxic potency, T helper 17 cells underperform, and neutrophils struggle to kill bacteria. Ultimately, the immune system becomes less efficient, particularly in the mucosal barrier, highlighting the essential role of a balanced microbiome in immune development. Inflammation of mucosal tissues is associated with causation, not cure of disease. This illustration contrasts the immune responses of the skin and the mucosal tissues. At the top, we see how systemic immunity defends the skin. Under normal conditions, healthy skin is protected by systemic immunity. But when the skin is wounded and bacteria enter, macrophages are activated and begin to release inflammatory cytokines. This triggers a classic inflammatory immune response, characterized by redness, swelling, and an influx of immune cells to the site of infection. Although the infection is eventually cleared, it leaves behind damage and distorted tissue that must undergo repair to restore normal function. In contrast, the bottom row demonstrates mucosal immunity, which operates in tissues like the intestine. Healthy mucosal tissue is protected by mucus and non-inflammatory immune mechanisms. When bacteria enter through the epithelial barrier, specialized cells like macrophages respond quickly, often without triggering inflammation. Local immune cells control the infection efficiently, while dendritic cells migrate to mesenteric lymph nodes to initiate a targeted adaptive response. Effector B and T cells, primed by the antigen, return to the infected tissue and eliminate the pathogen without causing significant damage. The infection is resolved with minimal disruption, and no tissue repair is needed. Unlike systemic immunity, mucosal immunity aims to contain threats quietly and effectively, preserving tissue integrity and preventing unnecessary inflammation. Intestinal epithelial cells contribute to innate immune responses in the gut. Intestinal epithelial cells are critical players in the gut's innate immune defense. These cells express toll-like receptors, or TLRs, either on the cell surface or within intracellular vesicles, allowing them to recognize bacterial components that breach the mucosal barrier. Once activated, TLRs initiate a signaling cascade that involves the translocation of NF-kappa B into the nucleus. Simultaneously, intracellular bacterial products that enter the cytosol are detected by NOD1 and NOD2 proteins. These receptors also activate NF-kappa B, further amplifying the inflammatory response. The activation of NF-kappa B prompts the epithelial cell to express a variety of inflammatory cytokines, chemokines, and antimicrobial molecules. These secreted signals recruit and activate neutrophils and monocytes, forming a rapid first line of defense against invading pathogens and maintaining mucosal homeostasis. Intestinal macrophages eliminate pathogens without creating a state of inflammation. This chart compares the characteristics of blood monocytes with intestinal macrophages and highlights how immune responses differ between the bloodstream and the gut. Blood monocytes are pro-inflammatory. They exhibit strong phagocytosis, killing ability, chemotaxis, and respiratory burst activity. They also engage in antigen presentation and produce cytokines, actively contributing to co-stimulation and the escalation of immune responses. Intestinal macrophages, on the other hand, behave quite differently. Although they retain the ability to phagocytose and kill pathogens, they lack chemotaxis, respiratory burst, cytokine production, and co-stimulatory functions. They also do not typically present antigens in the classical way, and their involvement in co-stimulation is minimal or absent. Phenotypically, the differences are even more pronounced. Intestinal macrophages lack expression of several surface markers seen on monocytes, such as CD14, CD16, and CD64. While blood monocytes express these markers robustly, intestinal macrophages are marked by a muted or absent expression profile, including low or no expression of co-stimulatory molecules like CD80 and CD86. These findings illustrate how intestinal macrophages are uniquely adapted to preserve tissue homeostasis in the gut. They clear pathogens silently, without causing inflammation, ensuring immune tolerance in a tissue constantly exposed to foreign antigens from food and microbiota. M cells constantly transport microbes and antigens from the gut lumen to gut-associated lymphoid tissue. This image shows a scanning electron micrograph of an M cell embedded in the intestinal epithelium. M cells are uniquely specialized epithelial cells that play a crucial role in mucosal immunity. Their primary function is to transport microbes and antigens directly from the gut lumen to the underlying gut-associated lymphoid tissue, or GALT. Unlike neighboring enterocytes, M cells lack thick microvilli and possess a pocketed surface that facilitates antigen uptake. By capturing and delivering microbial particles, M cells initiate immune surveillance and help stimulate appropriate immune responses. This transcytosis process is essential for maintaining the balance between tolerance and defense within the intestinal environment. The highlighted M cell in this image clearly illustrates its distinct morphology and function as a gatekeeper between the gut lumen and the immune system. This diagram illustrates the specialized function of M cells in the intestinal epithelium. Located within the follicle-associated epithelium overlying Peyer's patches, M cells are designed to sample antigens from the gut lumen. As shown, microbes and other particulate antigens from the lumen are taken up by M cells through transcytosis. These antigens are then delivered to immune cells within the intraepithelial pocket, including B cells, T cells, and dendritic cells. Dendritic cells capture and present the antigens using MHC molecules to activate T cells. This interaction results in the production of bacteria-specific effector T cells and plasma cells that secrete bacteria-specific antibodies. This pathway is critical for initiating adaptive immune responses in the gut and maintaining a balance between tolerance and defense against pathogens. Gut dendritic cells respond differently to food, commensal microorganisms, and pathogens. Dendritic cells in the gut play a crucial role in immune surveillance and tolerance. This image highlights their unique ability to extend long cellular processes, called dendrites, through tight junctions between epithelial cells. By reaching across the epithelial barrier, these dendritic cells are able to sample antigens directly from the gut lumen. This allows them to distinguish between food particles, commensal microorganisms, and harmful pathogens. Once an antigen is captured, dendritic cells process it and present it to T cells, helping to shape an appropriate immune response. This mechanism supports a balance between immune defense and tolerance in the intestinal environment. Activation of B cells and T cells in one mucosal tissue commits them to defending all mucosal tissues. When lymphocytes are activated in a Peyer's patch, located in the small intestine, they undergo a transformation into effector B cells and T cells. These activated cells exit through the efferent lymphatics and enter the mesenteric lymph node, continuing their journey through the lymphatic system. Eventually, they enter the bloodstream and circulate through the body. Because of specific homing signals, these effector cells are directed back to mucosal tissues, such as the respiratory tract, gastrointestinal tract, and urogenital tract, rather than to non-mucosal tissues. This specialized recirculation pattern ensures that activation in one mucosal site, like the gut, leads to widespread immune protection across all mucosal surfaces. It is a critical feature of mucosal immunity, providing comprehensive surveillance and rapid response to pathogens. Gut-homing effector T cells circulate through the bloodstream, searching for intestinal tissues. To home to the gut, these cells bind to the vascular endothelium using specific adhesion molecules and chemokine receptors. First, L-selectin on the T cell interacts with MAdCAM1 on the blood vessel wall. This is followed by stronger binding between alpha-4 beta-7 integrin and MAdCAM1, stabilizing the interaction. Simultaneously, the chemokine receptor CCR9 on the T cell responds to chemokine signals that guide it into the lamina propria. Once inside the lamina propria, gut-homing T cells are directed by chemokines such as CCL25, which are produced by the intestinal epithelia. These chemokines bind to receptors on the T cells, including alpha-E-beta-7 integrin, which anchors the cells through interaction with E-cadherin on epithelial cells. Together, these molecular interactions ensure that activated T cells efficiently home to and remain in the intestinal mucosa, where they contribute to immune surveillance and defense. A variety of effector lymphocytes guard healthy mucosal tissue in the absence of infection. Even in the absence of infection, the healthy intestinal epithelium and lamina propria are populated with diverse effector leukocytes. These immune cells remain on constant alert, maintaining a barrier of protection at the gut mucosal surface. Within the epithelium, intraepithelial lymphocytes, particularly CD8 plus T cells, are embedded among epithelial cells, ready to eliminate infected or stressed cells. Beneath this layer, in the lamina propria, multiple immune cell types operate as a coordinated defense network. CD4 plus and CD8 plus T cells patrol the area, while plasma cells secrete immunoglobulin IgA to neutralize pathogens in the gut lumen. Dendritic cells and macrophages monitor the environment and capture antigens, playing crucial roles in surveillance and antigen presentation. Mast cells contribute to immune regulation, and additional dendritic cells extend processes through the epithelium to sample luminal contents. This dynamic yet balanced immune presence helps maintain gut homeostasis and prevents pathogen invasion, ensuring that the mucosal immune system is primed and protective, even in times of peace. B cells activated in mucosal tissues give rise to plasma cells secreting IgM and IgA at mucosal surfaces. B cells activated in mucosal tissues differentiate into plasma cells that produce immunoglobulins essential for local defense. These plasma cells reside in the lamina propria, where they secrete both IgA and IgA. IgA, produced in the lamina propria, is transported across epithelial cells into the gut lumen. This process is facilitated by the neonatal Fc receptor, or FcRn, which binds IgA on the basolateral surface of epithelial cells. FcRn protects IgA from degradation and enables its transcytosis across the cell to the apical surface, where the IgA is released into the gut lumen. The FcRn receptor is then recycled back into the cell to repeat the process. At the same time, dimeric IgA is secreted by plasma cells and transported to the mucosal surface by the poly-Ig receptor. Together, these immunoglobulins form a critical part of mucosal immunity, neutralizing pathogens and preventing their access to epithelial cells. This efficient transport system ensures that antibody protection is actively maintained at mucosal surfaces, where the body is constantly exposed to external microorganisms. Secretory IgM and IgA protect mucosal surfaces from microbial invasion. In the mucosal immune system, plasma cells in the lamina propria play a central role by secreting two critical antibodies, IgM and IgA. These antibodies are transported across epithelial cells into the gut lumen, where they function at the front lines of defense. Once at the mucosal surface, secretory IgA and IgM bind to pathogens and toxins, neutralizing them and preventing their adherence to epithelial cells. By coating the microbes, these antibodies block their ability to invade tissues, effectively halting infection before it begins. The secretory component added during transcytosis stabilizes IgA, enhancing its resistance to enzymatic degradation in the gut environment. Together, IgA and IgM act as guardians of mucosal surfaces, forming a protective immune barrier that maintains intestinal homeostasis and shields the host from microbial invasion. Secretory IgA not only defends mucosal surfaces from external threats, it also plays a unique role in internal housekeeping. As IgA is secreted by plasma cells in the lamina propria, it can bind to toxins and pathogens already present in the tissue. During its transcytosis through epithelial cells, IgA escorts these harmful substances out of the lamina propria and into the gut lumen, effectively exporting them from the body. This dual function of secretion and detoxification highlights IgA's critical contribution to maintaining a clean, non-inflammatory environment within mucosal tissues. Two subclasses of IgA have complementary properties for controlling microbial populations. Immunoglobulin A, or IgA, exists in two distinct subclasses, IgA1 and IgA2. These structural differences give each subclass unique properties for microbial defense. In panels A and B, we observe the three-dimensional structures of IgA1 and IgA2, highlighting the longer hinge region of IgA1 compared to the more compact IgA2. Panels C and D break down the molecular components. IgA1 contains more O-linked carbohydrates and a hinge susceptible to proteolytic cleavage, whereas IgA2 is more resistant to bacterial protease due to its shorter hinge and additional disulfide bonds. Panels E and F show how both subclasses can join together through a J chain and secretory component to form dimeric IgA, the form secreted into mucosal areas. Together, IgA1 and IgA2 provide a complementary defense strategy, balancing sensitivity and resistance to microbial enzymes, thereby ensuring broader immune coverage across mucosal surfaces. What you are seeing here are the molecular structures of the two subclasses of immunoglobulin IgA1 and IgA2. On the left, labeled E, is IgA1. Its structure features an extended hinge region, allowing flexibility for antigen binding, but also making it more susceptible to bacterial proteases, especially in the gut and respiratory tract. On the right, labeled B, is IgA2. It has a shorter hinge region and a more compact structure, providing greater resistance to enzymatic degradation by microbes in mucosal environments. These structural differences enable each subclass to specialize in different microbial environments. IgA1 dominates in the upper respiratory tract, while IgA2 is more prevalent in the gut, where bacterial proteases are abundant. Together, they create a layered defense system, optimizing protection across various mucosal surfaces. Let's now examine the structural diagrams that highlight the functional differences between IgA1 and IgA2. In panel C, we see the IgA1 structure. It features a longer hinge region with O-linked carbohydrate side chains. These enhance flexibility but increase susceptibility to proteolytic cleavage by bacterial enzymes. Panel D illustrates IgA2, which has a shorter hinge region and incorporates N-linked carbohydrate chains near its tailpiece, enhancing its resistance to enzymatic attack, especially in the gastrointestinal tract. Panel E shows a dimeric IgA structure. Two IgA monomers are joined by a J chain, allowing it to be transported across epithelial barriers into mucosal secretions. Finally, Panel F reveals secretory IgA. This form includes the J chain and a secretory component acquired during epithelial transcytosis, protecting the molecule from degradation in harsh mucosal environments. Together, these structural adaptations equip IgA subclasses for site-specific immune defense across the body's mucosal surfaces. Let's take a closer look at how the two subclasses of IgA, IgA1 and IgA2, are distributed across different tissues of the body. In lymphoid-rich tissues like the spleen, peripheral lymph nodes, and tonsils, IgA1 dominates, accounting for 93% of total IgA, with a high IgA1 to IgA2 ratio of 13:1. This pattern is mirrored in the nasal mucosa. In contrast, mucosal tissues with greater microbial exposure show a more balanced distribution or even an IgA2 predominance. For example, in the colon, IgA2 makes up 64%, with a ratio favoring IgA2 over IgA1. Other mucosal sites such as the bronchial mucosa, tear ducts, salivary glands, and mammary glands maintain a moderate IgA1 bias, while the small and large intestines trend toward equal or IgA2 favored ratios, particularly in the ileum and colon. These differences reflect the specialized roles of each subclass. IgA1 is tailored for systemic and upper respiratory defenses, while IgA2 offers enhanced resistance to microbial proteases in the gut and lower respiratory tract. People lacking IgA are able to survive, reproduce, and generally remain healthy. Although IgA is critical for mucosal immunity, selective IgA deficiency is the most common primary immunodeficiency, and many people live without it. This chart shows the incidence of selective IgA deficiency across different populations, measured per million individuals. Saudi Arabians and Spanish populations exhibit the highest incidence, with approximately 6,993 and 6,135 cases per million, respectively. The prevalence is also notable among Nigerians, with nearly 4,000 cases per million, and U.S. Caucasians at about 1,667. Meanwhile, the incidence declines in other populations, just over 1,000 per million in Brazilians, around 250 in Chinese individuals, and fewer than 100 cases per million among African Americans and Japanese individuals. Despite the variability in prevalence, most individuals with IgA deficiency lead healthy lives, underscoring the adaptability of the immune system and the redundancy built into human defenses. In individuals with normal immune function, the majority of mucosal B cells produce IgA. This is evident in tissues like the nasal glands, lacrimal and parotid glands, gastric mucosa, and small intestine, where 69 to 82% of B cells produce IgA. But in individuals who are IgA deficient, the immune system adapts. In the absence of IgA, there's a compensatory increase in other antibody isotypes. IgM production rises dramatically across mucosal tissues, reaching 75% in the small intestine and over 60% in the gastric mucosa. Similarly, IgG becomes more prominent, especially in the nasal glands and salivary tissues, where up to 57% of B cells express it. This shift illustrates the immune system's remarkable plasticity. Even when a dominant antibody class like IgA is absent, other isotypes step in, helping maintain mucosal immunity and preserve host health. Th2-mediated immunity protects against helminth infections. Helminths are parasitic worms that fall into three major groups: roundworms, flukes, and tapeworms. Roundworms, or nematodes, are responsible for a variety of serious diseases such as ascariasis, dracunculiasis, also known as guinea worm disease, elephantiasis caused by lymphatic filariasis, and other common infections like pinworm, hookworm, river blindness, and trichinosis. Flukes, scientifically known as trematodes, include parasites that cause schistosomiasis and fasciolopsiasis, both of which affect the liver, intestines, or bladder depending on the species. Tapeworms, classified as cestodes, lead to tapeworm infection, often acquired through the ingestion of undercooked meat or fish containing larval cysts. To combat these parasitic infections, the immune system relies heavily on Th2-mediated immunity, which mobilizes eosinophils, mast cells, and IgG to orchestrate targeted responses and expel the parasites from the host. A protective Th2-mediated immune response is essential for eliminating helminth infections and minimizing tissue damage. Key features of this response include a strong parasite-specific IgG production, which arms mast cells and eosinophils for rapid degranulation upon antigen encounter. Mucosal mast cell hyperplasia is a hallmark of this response, enhancing the release of histamine and other mediators that contribute to the expulsion of parasites. Eosinophilia in both the blood and intestinal tissues reflects the recruitment of these potent cytotoxic granulocytes, which play a central role in attacking helminths. Changes in intestinal muscle contractility, along with goblet cell hyperplasia and crypt hyperplasia, promote mucus production and increased motility, mechanisms that physically dislodge and clear parasites from the gut. Villus atrophy, a consequence of inflammation and immune activity, reduces the absorptive surface area. This, in turn, may contribute to weight loss, a physiological cost of mounting a strong Th2 response, but a necessary trade-off to restore intestinal homeostasis. During a helminth infection, naive CD4 T cells differentiate into either Th2 or Th1 effector cells. The Th2 pathway provides the most protective benefits against helminths, while the Th1 pathway can lead to harmful tissue damage. Th2 cells produce interleukin-13, which enhances epithelial cell turnover and increases mucus production from goblet cells, helping to flush out the parasites. They also produce IL-5, which activates eosinophils. These eosinophils release granules that kill the helminths directly and contribute to host defense. Th2 cells also drive B cells to switch to IgA production. This parasite-specific IgA binds to mast cells and triggers degranulation upon subsequent exposure to the parasite. This immune cascade further amplifies expulsion mechanisms. Meanwhile, Th1 responses are less effective and more damaging. Th1 cells secrete interferon gamma, activating macrophages that release inflammatory mediators. This leads to tissue destruction rather than parasite elimination. In the context of helminth infections, a Th2 dominant immune response is critical for protective immunity with minimal host damage. In contrast, a Th1 dominant response may fail to eliminate the parasite and result in significant inflammation and tissue injury. The mucosal immune system is uniquely adapted to maintain immune balance in areas constantly exposed to the external environment. Anatomically, there is an intimate relationship between mucosal epithelia and lymphoid tissues. These include organized structures such as Peyer's patches, isolated lymphoid follicles, and tonsils, as well as more diffuse lymphoid compartments. Specialized antigen uptake mechanisms, especially those involving M cells, facilitate immune surveillance in these mucosal regions. In terms of effector mechanisms, activated effector T cells are present even in the absence of infection, offering a rapid response capability. Plasma cells are strategically located within the tissues where antibodies are needed most, enabling immediate local action. The immunoregulatory environment is characterized by an active suppression of unnecessary inflammation. This helps prevent overreaction to food antigens and other harmless environmental exposures. Mucosal macrophages are inflammation anergic, and dendritic cells promote tolerance rather than activation, maintaining a peaceful coexistence between the immune system and the vast microbial and dietary antigens encountered at mucosal surfaces.